Oh, hello, everyone. I'll be talking about a small project that I've been working on recently, and that was funded by a HEAS seed grant. And so where we've been trying to confirm MPS, which I'll explain what it is in a second, in the medieval population of Potterbrunn. So we've been working on this in a team of 4 people. There's myself, who's been working on the lab work, essentially. Daniel Fernandez has been doing the bioinformatics work. Marietta Schlein-Nicola was the physical anthropologist who found those skeletons in the first place. And Adrian Daly, who's a medical doctor working on inherited diseases. So MPS, what is it? It's actually called, in its long form, mucopolysaccharidosis. And it's a metabolic disease of involving problems in lysosomal storage, so metabolites being processed improperly. It comes in about over 12 different types with different manifestations, and it causes death quite young. In the US, about 1 in 25,000 births is affected by mucopolysaccharidosis, but it's not necessarily severe. Sometimes it's attenuated and doesn't really show up as a disease. But most importantly, it's transmitted in an autosomal recessive fashion, which is crucial for our purposes. So the symptoms of MPS is a lot of neurological symptoms, mainly motor and sensory function problems, which depend on which type of MPS you're dealing with. There's very distinct facial features. There's certain types of dwarfism associated with it, and respiratory and cardiac issues ultimately cause death. And on the skeletal aspect of things, so the skull shows hydrocephaly and an enlarged mouth and nose. The hands tend to be small and characteristically shaped. The torso is much smaller, which is the cause of dwarfism. And in general, bones tend to be of the wrong size throughout the body. Looking at the actual population that we're looking at in this study, we looked at the medieval population from Potterbrunn, which is in Lower Austria, not too far from Vienna. It's a 9th century population, and 199 individuals were excavated from this particular period there. Of these 199 individuals, 9 were identified by Maria as suffering from potential MPS. These were all children ranging from 9 months to 12 years old, and they had MPS at variable severity. So on the right here, you have the bones of one individual suffering from MPS. Which type of MPS did they suffer from? It was possible to narrow it down to 2 potential types. One is MPS 1, which is also called Hurler syndrome, and the other one was MPS 4, which is Morquio-Brelford syndrome. Why do we really care exactly which type they are? It's because genetically, each type is associated with a particular mutation, or a particular set of mutations in specific genes. So we know that we're looking for mutations in 3 different genes. One is GLB1 in chromosome 3, IDUA in chromosome 4, and GLNS in chromosome 16. So knowing this, what were we actually looking for in these individuals? Well, obviously our dream aim would be to identify the exact mutation that is causing the disease in this population. But this is extremely challenging in ancient DNA. Even in non-ancient DNA, it can be difficult. So a more realistic aim for this small study was to identify family relationships between the diseased individuals. For this, we looked at 7 teeth, 6 of which I'm showing you here. Because they are children, all the teeth were deciduous teeth and were very small teeth, but that was enough for a DNA study. In a very classical DNA study way, we cut a root of each of these teeth, extracted the DNA, prepared the DNA for sequencing, sequenced the DNA as much as we could in this particular case, and then analyzed the data that we obtained. The first thing we looked at is whether we did get any human DNA in the first place. The DNA preservation in terms of percentage of endogenous human DNA we found was not particularly great, a little bit disappointing to say the least, for medieval populations from Austria. The highest being 3.44% endogenous, and the lowest really had barely anything at all. What was even more disappointing was the fact that all of this DNA that we did find turned out to be contamination. So first there was suspicious damage patterns on the molecules that we extracted. The read lengths were a bit weird, and then we ran some specific bioinformatics software, this Calico and Carrier RYIX, and it gave us the defined answer that it was all contamination. So where was this contamination from? The first thing we looked at was whether it was contamination from the actual lab work, which happens sometimes. Luckily, this time it wasn't the case. So we ran controls with the samples and these came out clean. So it must have been contamination that was on the teeth before they came to us. This can have come from a range of manipulations that happened in the past. And it was confirmed that this because we had several mitochondrial haplogroups that we discovered on those teeth, which were obviously not the ones of the individuals the teeth belonged to, but of different people, because there were different haplogroups found there. So the contamination must have happened throughout time. So what do we do next to actually try to get some information out of those? First, we'd like to get deeper in the teeth to try and get away from this contamination that's probably on the surface of the teeth. So to sample in the crowns. Unfortunately, these teeth are very small, so there's not much to play with here, but we'll do our best. And if we could access the petrous bones, that would probably help us as well, because not only does the petrous bone tend to preserve the DNA a little bit better, but it is also deep inside the skull, so probably less handled and less prone to contamination. So hopefully we'll be able to identify a mutation that causes the disease in this population. But in the long term, what would be also really nice is to look at the rest of the population, because since it is an inherited disease, it'll be very interesting to see what patterns of inheritance it has throughout the population. And if we find attenuated cases, for example, which were not identified in the skeletal through the skeletal morphology. So thank you.